Well, thank you very much. It's just a pleasure to be invited. It was sort of, Alan said, hey, you want to go to dinner? And I said, all right. I didn't know who the heck I was going to dinner with. And I happened to be Larry, so they ganged up on me, and here I am. So I want to welcome all of you to Madison. I went to grad school here. I went to Berkeley, and then I came back. I really fell in love with the place. And in particular, from my arts background, Madison really supports the arts, and there's actually this cool law here is that 10% of every new building, of the funding to new building goes into an arts project. And so I found that very interesting because I grew up in Kentucky where none of the arts were really supported as much as I've seen here in Wisconsin. So I'm going to get started and then go through sort of to talk about my life, but I wanted to start out with a cat, because I realized, this is my cat Bonsai, is that, this was going to be on YouTube, is that in order to get a lot of hits, I probably should put a little nice cat up there. Anyway, so if you see my cat, give it a click, thumbs up. Anyway, so this is the title of my talk, Too Creative for Science. And so it's probably sort of an odd, for this crowd, it's probably an odd sort of title. So I took this, when I was talking with Larry over dinner, he said, this is the title. This is good. So I had told him a story when I first started in grad school that I started out in a lab, and my mentor at that time, he knew I had this art background, but he said to me one day, and I was probably about 4 months into my graduate work, and you're at that time where you still feel like an undergrad and you sort of agree with everything your professor says, so on a via, and he said to me, you're too creative for science. And I was sort of taken back, and I was like, what, And I left for 2 days, and I didn't come back. It sort of shocked me out of myself, and I was at that point where maybe, is this really what I want to be up against, constantly battling, people like this that don't appreciate the arts in the sciences? And I realized that wasn't the right environment for me. And so I ended up switching labs because of it. Just this one little statement that sort of changed it. And I found someone who really appreciated the side of me that is creative and artistic. And that has to do with imaging, so I'll talk a little bit about that. Anyway, so that's where the title comes from. So as I began to sort of think about, is it just me who's too creative for science? How could this be? How could this professor say this to me, So, of course, many of you know Da Vinci is probably the true scientific artist, right? So not only an artist, but, had studied anatomy. And my father, from my own personal experience, had done the same thing. And so it seemed mind-boggling that someone would say that to me. And probably one of my other favorite scientific artists is Ernst Haeckel. If you do a Google search, you'll find these amazing pictures. This is of fungi. This is back in the late 1800s. Amazingly huge amount of creativity, imagination, and skill to draw and to tell people about what he was seeing in the lab, in nature, and stuff like that. And he is actually really instrumental in the adapters of the theory of evolution. And probably one of the famous pictures you've probably seen is this comparative embryology picture here. And so what this really shows is that there was, art's been used for a lot of time, and creativity to support a lot of scientific theories. And so what Haeckel had shown is that early embryos are more closely similar in related species. And so these are a hog, a calf, a rabbit, and a man. A lot of the early embryos look very similar. So this was it made people believe that maybe this is true, this evolution, we all arise from, a common ancestor. And another thing about Haeckel is if you just go through the stacks and stacks of images, it's absolutely beautiful. And I think for coming from an artistic background, it's stunningly beautiful, amazingly intricate. It's just, I can look at it all day. I just absolutely love it. And of course, probably the other sort of thing where I found that art and design was used to advance science is the case of the discovery of DNA. So many of you know about the story of Rosalind Franklin. She did this X-ray diffraction picture. Of course, this is a 2D image, right? So in order to come up with the structure of DNA, how was it done? And of course, in collaboration with Watson and Crick, they made a 3-dimensional model. And so that model, bringing that up to that level, really made them visualize and to see where those bonds were. And so that, so I saw these things that, in these 2 days that I left lab and I was realizing whether I was going to come back, I realized he was wrong, my advisor was wrong, and that there was a lot of cases where art and creativity and using that was important for discovery. So in speaking with my dad was a professor for over 40 years, and he knew the trials and tribulations of being a faculty member. So this was something actually was in my house a lot. So he says, you always know that this quote is that great spirits often encounter violent opposition from weak minds. And so I just sort of stuck with that and realized is that keep doing what you're doing, and you'll end up with greatness. So I'm going to sort of split up my talk into talk about my family, my background, a little bit of my science, my teaching, and my outreach. So here's my genetics. So there's my crazy father there. My father was my mother's advisor, grad student advisor. So she was a ceramist and art teacher. And then I'm the oldest child, so that's me on the left there. My sister Tarsha is a graphic designer. She's worked for Paul Mitchell products. She makes a lot of shampoo bottles. You've probably seen them. My other sister Zesha works for a company, a design company that does a lot of bottle and package design for Procter & Gamble. And my brother is an industrial designer who makes things from tools to motorcycles, anything. He's done a lot of things. And he had worked in China for the Procter & Gamble market, making Crest boxes, or how do you package things how do you package gym shoes in boxes and make them cheap, send them across the Pacific. But that's my family, and so a lot of people always how did I end up a scientist? And one thing about my father is that he also was a medical illustrator. We worked part-time when he was a faculty member. So to me, it never, my parents really loved science a lot. My mother was, in addition to taking her ceramics and art and drawing classes when she was a graduate student, would make jewelry out of cross-sections of animals she was doing in her biology class. So my dad often told a story is that she brought in a baby shark to my father's studio. It was dead. She was so excited about the designs that she saw in the cross-section. So this was sort of my life growing up. And so I didn't really see any difference between it. But as I got out into the world, a lot of people thought it was a very strange background. How could I be what I be? So my father was a pupil of Maestrovic. So has anyone seen these? This is down in Grant Park in Chicago. These are statues by Maestrovic. So my father helped cast these. So Maestrovic was the pupil of Rodin. So just as an Indian lineage where people come from is very important. So the philosophies have gone down from generation. And so this was Maestrovic. So I grew up knowing about him, his philosophies, and how it sort of changed my father's being. My father had got a full ride to Syracuse where he had been a faculty member and then followed him to Notre Dame. So this is my father can do just about anything like Da Vinci, he can do just about anything. He was a painter, a drawer, sculptor, and stuff like that. That's a self-portrait of my father later in his life. Here's my father sculpting in the studio in our house. This is, if you know anything about football, this is Ben Schwartzwalder, was the Syracuse football coach. My dad played football at Syracuse with Jim Brown. Schwartzwalder was the head coach at that time. So we had interesting people coming into our house when I was a kid. So he was doing the bus that is actually up in the Carrier Dome. It's in Syracuse. That's probably, what year is that? It's probably in the late 80s in my house. And this was a studio that was built on the side of my house. There were a lot of people coming in that were, I didn't really see boundaries and things. There was always someone interesting coming in to speak to my father. One of the other people that I met growing up was Don Chandler, who was the sculptor for King Kong and Jaws. And so he actually worked with my father at Syracuse under Maestrovic. And so he was asking my father how to build the armature for King Kong. So you realize there's engineers and artists and sculptors all involved in that process. And so that was really something neat to meet these kind of people who were coming in constantly and using the ideas, the arts, and the philosophy on building these things, you know, for the movie industry. So this is a picture of me. There's me at 2 years old. This is 1974. These are the kind of people I grew up with. So these are artists, you know, hippies, musicians, they're philosophers. They all came to study with my father. So it was always a very lively home. It wasn't, you know, we grew up in northern Kentucky. We didn't really lock our doors. So there was always 1,000 people around our home. And there were always people wanting to learn. So I was in an environment of constant barrage of people interested in 1,000 things. So boundaries really weren't there for me growing up. And so here's my home. It's a English Tudor home. So this was built by an artist, Julian Bechtold. And the picture of ivory there, so in my home was the very first carving of the modern ivory soap bar done by Julian Bechtold. So there's a lot of history here. And it's actually a very unusual house for Kentucky, and so it's a historical site in Kentucky. And then my dad built on the studio in the back. So this is as you walk in. There's sculptures that my father had done in wood of Maestrovic. There's reliefs as you walk everywhere. Just wanted to take you on a tour. On the front lawn, there's giant sculptures. Very unusual. People thought we were the Addams family at some point. So we were just sort of up on the hill, and we were these crazy people with sculptures in our town, you know. And it just was a very interesting place to grow up, and it's something I've never seen. This is the part of the studio that my father made. More reliefs. This is the backyard. So my father was a jack of all trades, sort of like Da Vinci, so he can do woodworking, carving, welding, and he had actually built all the cement work in the backyard here. And those little sort of steps there were always a lot of quotes around my house to read all the time. And one of them that always stood out to me besides the other Einstein one was this one about Picasso, is that every child is an artist. You know, the problem is how to remain that when you grow up. And so I always sort of had that in the back of my mind as I was going on to graduate school and thinking about really what appealed to me and what I wanted to do. And so I'm going to talk a little bit about my science. When I started my undergrad, this is the first sort of image I saw. This is if any of you know, this is a DNA gel. To me, it's not very exciting. It's a bunch of bands on a screen. So is this really biology? Is this life living? Is this what I was going to be doing? In my undergrad, I ran a lot of these gels. And I just felt like I wasn't able to grasp science with this kind of technology. And then in my developmental biology book, I stumbled upon a picture like this, right? You can see the stark difference. For me, I had looked as if I saw a Picasso. I almost fainted. I'm like, oh my gosh, what is this? So this is a confocal image of an early C. elegans embryo. The way that you're seeing this image is through the invention of a confocal microscope, which actually ended up being my mentor here in Madison, John White, which I'll talk about. So here's John. So when I met this man, so after I moved to my other lab, I moved to John White's lab. John White, the inventor of this microscope, really loved the arts, was very supportive of it, and I felt like I had the right environment. And one of the quotes that I always loved, and I had been struggling, I'm dyslexic, so I'd always had trouble taking tests in class. But I was very good speaking, I was very good doing projects by myself. So he said, don't worry, this is what his quote was, don't worry, I got a D in physics, but I invented a microscope. And I said, all right. He's got like the Queen's Award for best invention. I was like, oh, right. He just was the most humble man. But it was that one thing that I said, keep plugging away. There are people who, you know, you can get any grade. Grades don't matter. It's really what's inside of you. And so that was like, we were two peas in a pod. So that really changed my life. And since then, I've been sort of completely enamored with microscopy and imaging. And I'm just going to show you some images that I find very beautiful to you, but that have to do with things I see every day. So these are just pictures from around the world using this kind of microscopy. So 2 cells dividing. These are neural stem cells. I can look at these things all the time. It's just absolutely stunningly beautiful. This is something recently came out, any of you have seen it, is this brain bow. So they're able to label each different cell type in the brain. I almost peed my pants when I saw this. I was like, oh my God, I got to meet this guy. I mean, it just was really stunning. Just the technology and the creativity of using different fluorophores so that you can label different cells. It just was amazing what we can see at this point. So these are things that, just imagery for me, coming back from that gel, imagery really, I feel like I can understand biology more. It's a part of my life. I see it as beauty, and I see it as something I want to figure out how it works. And so the work in my lab, just going to briefly show you, is understanding cell division. So how do you go from one cell to two? So, and as Alan has said, errors in this cause cancer, birth defects. So if you don't, trisomy 21 is an example of something where cell division has failed. And so we want to understand the basic process of how this occurs. We use a lot of video microscopy in my lab, and we tag proteins with GFP from the jellyfish. So you're able to see, this is the mitotic spindle and the DNA segregating in the cell. And this is done with confocal and another microscopy called multiphoton microscopy, which allows embryos to develop a lot longer and not die. So when I started studying cytokinesis, I'm always very fascinated with also the history and where I am in that point in history to try to understand where people were, the questions they were asking past, and what questions should I be asking now? So I went back to find this. And John, my advisor, was very much into this. I went back in time and I found that Walter Fleming was one of the first guys to do this. So he actually sketched the first thing. So sketching and being an artist like Da Vinci was very important to understanding the process of mitosis. He coined the term of mitosis, and he discovered this thing called the midbody, which is how my work started to get going. So this is one of his pictures from 1876. And my advisor, John, had said, today the detail, and it's absolutely miraculous, and it's amazing, because my advisor had said that this is probably a microscope with a lens that was a cut end off of a beer bottle. Like it was that crappy of glass, but they were able to see this. So it was amazing what scientists were able to do and to painstakingly draw these things to show people what was going on in the cell. And you can see from pictures today, it's not very far off. Sometimes these pictures are really exactly what they had in the late 1800s. So it was worked on for many years, obviously over the years, but there was a big question of, still the question is, how does a cell divide? And in 1891, when he discovered this structure called the midbody, the midpoint of the cell, he had pointed out in this picture here is that all cells have this structure. And that he said this structure is very similar to the cell plate in plants. And so probably many of you know that in your textbooks that plants and animals divide differently, right? But in the 1800s, that wasn't the case. They had said that they could be the same. So the mechanisms to build a cell plate in plants is probably the same thing happening in animal cells. And people poo-pooed that idea all these years. So I came along, I decided to revisit this kind of question. So what is in this midbody? Are there things that are building the cell plate in plants like in an animal cells? So this is in a mammalian cell, and this is when a mammalian cell divides, it divides by crawling. And so it's going to crawl away from each other, and inside of that intercellular canal is the midbody. And so this is what you see inside. That's the midbody. So what I think was going to go on here is, if what Fleming was right, things that were in this structure are important for cell division, maybe we can identify all the proteins in there. So that's what I did. So what I did is isolate them. So these are midbodies from a Chinese hamster overline cell, and so we isolate tons of them biochemically. There are a lot of they look different at different times in the cell cycle. The one in the middle is actually cells that are growing culture, actually aneuploid, so you can get tetrapolar spindles, but all of these things contain the same component. Then what I did, so I don't have to bore you with technical details, is we took these and isolated the protein sequences from everything in the midbody. So this came up to be about 500 proteins. So, and 100 of them were new proteins never found to evolve in cell division. And the big surprise was that a lot of them were very similar to membrane trafficking proteins, which are exactly what they are found in plants. So that's sort of what I became known for, is that plants and animals do divide similarly, and this structure is very similar to the cell plate in plants. So to test the proteins that are here, one way that I do that is I use C. elegans. So I go from mammalian cells into C. elegans, and this is a movie of the C. elegans worm. And it's about 1 millimeter long. And inside the worm are little embryos. If you could see the little embryos in there, it's hermaphrodite. If you cut the worm in half, we take those little embryos, which are about 30 microns by 50 microns in size, and we get a video like this. So this is right after fertilization. The egg meets the sperm. And if you can look at the time there, in 15 minutes, you're going to have your first division. In human cells, the division is going to be about 24 hours. So using this model organism is very useful because it's very rapid. The other thing is these embryos are transparent. And they can go on into, you can watch and watch until they eventually hatch. So we use a lot of video microscopy, a lot of gigs and gigs of data as well. But one thing I think is important in my lab is also how do you portray this in a two-dimensional form, because you can't always do this in a journal. So learning, when you join my lab, you have to do Illustrator, Photoshop, all these kind of programs. ImageJ is something else we use a lot to turn over our images, but portraying the development through time. And so this is just past that video is looking at the embryo all the way until it hatches into this young worm. So then the other thing is what we do now is go back to this GFP, we knock out the genes that are identified in this midbody and see what happens to the normal development. So I'll show you just one example of that. So here's RAC1 was a mammalian protein. It also had a homolog, similar protein in C. elegans. If you watch this embryo, so it doesn't have RAC1, the cell looks like it pinches in, but it fails. And the two nuclei come back together. So this would be like a tumorous cell. So this is very exciting. This is like the first day of my screen. So we'll go back to that. So this mitotic spindle will set up. It looks like it's going to pinch in, and then it's going to pop back up. So think of a balloon that you're twisting, and you need to insert new latex there. Maybe that latex is not there anymore, and it pops back up, right, when you're making those balloon animals. This is what's happening in the cell. So this protein is required for that final pinching off of the cell. And about the 100 proteins that I identified, most of them have a similar phenotype to that. So I have all these new proteins required for this division event. So hopefully down the road, clinical scientists will take this information and use it to make drug targets for cancer. So that's sort of the big picture idea. The stuff that we do later, so here's another thing, is we'll do fixed images in human cells. So this is just another beautiful image of something identified in that midbody where it localizes throughout the cell cycle. I always sort of love this quote, is that, at least for me, is that the things that I do, I really delight in its beauty, and I think I can look at it all the time. And I think if you have a perspective from me as an artist to appreciate that beauty, I see things differently sometimes than I think my students sometimes do because they have a different background. But I appreciate people's backgrounds, and everyone, we come together and having that kind of discourse is like we appreciate the beauty in it. Otherwise, we wouldn't do what we do, I think. And I probably wouldn't be in science if I didn't see these kinds of images. I really would be turned off. So now I'm going to switch to a little of my teaching. And so I always thought this quote was very good. Is the only thing that interferes with my learning is my education. And being dyslexic, I always felt this was very true for me because I always had trouble with taking those tests and working hard. But everything else was right on. And I saw a talk by Ken Robinson, which probably many of you have seen, is that the education system is educating people out of their creativity. They're taught to, this is back from the Industrial Revolution where people have to do certain things a certain way, and that's it. And so as I was creating my course, I came across this other Einstein quote, and it says, I think, most teachers waste their time by asking questions which are intended to discover what the pupil does not know, but the true art of questioning is to discover what the pupil knows or is capable of knowing. So that the potential, every student has a potential for success. You know, I think that in my philosophy, even with my father's teachings and things I'm thinking about science and my own struggles in science, is that I think it's like that's where education should be. So I created this course. It's called Genetics 677. You can do a Google search and find it, but it's an active learning course. So I didn't want to go in and lecture. I find lecturing, I did lecturing for my first couple of years, I find it boring, and students are not responding. So I've taken a lot of courses on active learning. What the students do is they learn about a gene in human disease. It's of their own choice. A lot of it's from something that someone in their family suffers from. They find the associated gene and study it. And the other thing I do is I have them learn to give presentations and design slides. So how do you market yourself? How do you give a good presentation? That's really important to do. And the other thing they do is they designed a website using Weebly. I don't know if any of you know Weebly. Weebly is just a sort of freeware web-based program to create websites. And I found it was very useful to drop and drag stuff for students. And so the other website is Wix that some students in my lab use, but Weebly is generally pretty easy. And so by the end of the semesters, the students have published work. So the first day in class, I always tell them the reason why we're doing this is I don't come to work and fill out bubble sheets. And I come to work to think and be creative. And we want to get away from sitting and memorizing stuff. I want you to really think about that. And a lot of kids sort of freak out a little bit because they're ready to memorize because they're taught to memorize. I said, I don't have bubble sheets stacked up in my office. I'm not filling these things out. So, and the other thing I tell them is that everyone from day one has an A. And I think this is sort of a reverse psychology. So back to that one quote is that everyone has the potential for success. So the only way to, you lose points by not doing the work, not doing the thing. So I think students really appreciate this because they realize that they're worth something. Every person's voice is worth something, and they're willing to give those things. And I think it opens up the ability for them to just branch out into a different way of thinking because now they don't have this thing over their head. I need to get A. I need to get A. You know, none of that kind of stress is in my class. So, and the textbook that I use, probably many of you have seen, is Presentation Zen. So I don't have a science textbook. I use journals from the web. Gar Reynolds, one of my idols of creating slides. So this is an optional textbook for them. A lot of them do get it. But I tell them that I use this book as well on learning to give talks, how to set up slides. And I have a speaker come in and learn how to set up slides and how important that is of trying to remove text from slides and maybe more imagery. And they're there to come and listen to you, not there to read. So I mean that's something they're going to be do when they graduate. They're going to go out in the world. They want to listen to you, and so I try to really get that out there. And so the other question I sort of make once we go into the lab course is I have them raise their hand. How many students have made a website? And maybe one raises their hand, and I'm sort of like, what? I taught myself how to HTML when the internet first started. Like I was actually sort of amazed that they don't have that curiosity to learn what they're using from day to day. So that actually was the biggest surprise I had when I started this kind of concept. And so the first couple of weeks is a complete struggle for these kids to try to put together, why am I doing this website? Why am I doing this? This doesn't make sense. You know, it's like, but it's learning. But this, I'm going to show you the results of this. After a lot of hair pulling, this is what happened. So this is student Jessica Keek. She's one of my best students. She chose the gene for breast cancer BRCA1. And, you know, they can create, Weebly's got these sort of standard templates. And so they create sort of like a Wikipedia page on this gene and disease. And so, and everything on the side there, you know, they talk about the gene, the protein, conclusions. And the other important thing I wanted to point out is they propose at the end of the semester where they think researchers should go with this work based on what they found with online materials. So this is all free out on in the web. Your parents can see it. All these kinds of all this information that taxpayer money pay, you can access that too, and you can come up with an idea. So that's all included in that. And so that's the project. Here's another project I like. A lot of humor, creativity. A lot of students ask me, they say, what are you looking for? I'm like, I'm not looking for anything. I'm looking for what you think is important. And so it's a struggle for them, but they eventually get it. But including humor, what on a website is going to pull people in to look at your project? This one, he had a family of, all his fathers and uncles were all going bald, and he was all petrified about it. So he wanted to know more about it. So his is very funny if you see it. Here's another. So here I've had students with dyslexia like myself in my class. So she struggled. These are all senior genetics students. She had struggled in all her classes. She probably had B's and C's. Of course, she chose dyslexia and a gene associated with it. And I was impressed, and she had one of the highest grades because she had freedom to do what she was best at. I thought it was just very moving to see that. And the other thing I wanted you to point out at the bottom is we put maps, the hit maps of where people are looking at. And so this is a great, you can look, this is just from the other day. This is my first year class. People are still looking at their work. And when kids started to see that other people were looking at their work, their writing was improving. I didn't have to worry about the writing. But they were realizing is that what they are producing is worth something to the world. I think when you put out these websites, that is something I did. Probably half the class did the first year. Now everyone does it. And everyone's more excited who from what country is looking at their website and why. And then there was a Thai student in my lab, and his dad lived in Bangkok, and he had hit everyone's side in the class, and everyone was excited. But it just, for me, that little map made all the difference for the kids. It really was like, this is not a project that Anna's going to look at. It's a project that everyone's going to look at. So I feel, the kids feel really fulfilled about this. They incorporate YouTube videos. So this one's about narcolepsy. This is a narcoleptic dog. I have them include everything. They're always sort of nervous. Can I really include those YouTube videos or not? Yes, why not? There's interviews of doctors who work on Alzheimer's on there. There's a lot of information in there. But these are things that are important for the students that they think people should know about their gene and disease. So it's a no-holds-barred thing. And they take things from public databases. And so here's an example, someone this semester working on celiac disease. So they have their protein, they find proteins that interact with each other, and then they propose what they think is what they're going to do down the road, what they're going to work on. So which has never been done. So they leave it there at the end of the semester, and they talk about that. I've had a student who was in my first year class working on Huntington. He's now in medical school. He goes, I got an email from some guy in Taiwan. He says he wants to collaborate with me. And I said, really? He says, yeah. He said, I found something that he didn't know existed about Huntington's disease. And this is on these websites. And these kids are like beaming. These are like 2 years later is that they're doing something that's worth something. And so I'm really, really proud of this course, and I feel like it's really been, the kids just absolutely love it, partly because there's no tests. I think that's one thing. There's that stress of the test, but you have an A starting out, and it's amazing what you can actually see the kids do. And a lot of professors are like, you don't lecture and you get this? How does that occur? They don't understand how I can produce something like this. And the kids also give their final presentations about their work. And most of the faculty who sit in are just pretty stunned at the results of having discussions on Tuesday and the lab on Thursday. So that's sort of how I've brought in, I want people to be creative. And this is where I sort of felt science to me this made science easier for me to understand from someone who's dyslexic, maybe I have a hard reading, but doing projects is fun. It's finding out answers to things. I really enjoy it. So that's my teaching, and I was going to move a little into my sort of my outreach that I've been doing with science and art. So keeping my genetics expression of art very high, things that I've been doing. So in the building, in the genetics building, when I first came here, there was an empty wall, and coming from a household of 1,000 pieces of artwork on the wall, I can't sit in a room with white walls. So I went up to the dean's office, my first year, and he said, they'll give me 15,000. And I did. So that's what I did, images that are taken from scientists in my building. And now it's a tour on campus. Kids come to see it, and they see what science is being done and how beautiful it is, but also what is it and what does it mean? It creates a lot of dialogue. So I've been very proud of that. And that sort of led to this this scientific art show that a lot of people on campus helped me with. So I gave another talk about science and art, and this is what came out of it. My dream is really to have a scientific art museum in town, but that's still being worked on. So this was, a collection. This was initially opened at the Dane County Airport, which you've come through. There's little display cases, which are down here. And just sort of collected tons of artwork. And the printing press that's on campus that works with artists printed it up on really nice paper. And the former Chancellor John Wiley, paid for all this, put up there. So it's been the most popular show at the airport I think they ever had. It was up for over a year, which was unheard of, but they had more people looking at it. So, and then we've been contacted by PBS and USA Today, and it was pretty exciting. So, and it's been touring around campus, and it's going to go to the NSF, and a lot of people around the country want it now. So I've been very proud of this, and I think it's really cool. I think people are initially drawn to it because it's beautiful, but then they allow them to ask questions once they see that. What is that? What does it mean? So that picture up there on the right is, when you touch a leaf and it's fuzzy, that's a trichome on top of a leaf. That's the fuzziness on a leaf. So, that's the thing, is you can make science, accessible to a lot of people through the arts. I think kids immediately are drawn to that, and then you start to tell them, and then they start to ask more questions. So I met this first grade teacher, Troy Dassler, has been in my lab for the past year. He's actually now teaching at the he's going to be the learning and resource educator at the Wisconsin Institutes of Discovery. But Troy's been interested in video microscopy, so a lot of things I do in my lab for his first grade class. And so he had got a grant from the NSF for these microscopes that are about $200 at the time. I think now they're even cheaper than that, $100, that students can actually see images, anything they can pick up from nature and put it onto there. He's actually probably the most amazing first grade science teacher I think I've ever met. He's probably the most popular science teacher in all of Wisconsin. I know people at the NSF absolutely love this guy. He's given a TEDx Madtown talk, so you'll see some of the things he's been doing in his thing. But one of the things we've been working on is a children's book, and those are those little plates I showed you. So those are the pictures of that, so I'll show you the video again. And so what we're doing is printing the bacteria that the worms eat, and it's going to be sort of an ABC book for first graders, and try to introduce them to the model organisms, C. elegans, and the plates. But this is sort of a couple days of working with Troy. We came up with this technique where we went to Michael's and got some letters and dipped it in bacteria and grew it overnight. Voila, we got beautiful letters and bacteria. And we can let worms grow on it, and the bacteria disappears over time. And so we're doing these montages and stuff. So we're really playing around with it. And this is my student, Kelly, who's very much into outreach. She worked at the Atlanta Science Museum, and so she's all into helping with this. So, yeah, we're taking pictures of the plates and worms. And so hopefully down the road that project will be finished, but it's continuing. And Troy, Troy came to my office one day, and he's like, we're doing the letters, and then he came and he painted me in bacteria. And Troy's like, I'm not very creative. And I'm like, what are you talking about? He's amazing, So, it's just in the lab, the interaction with someone like Troy and my students and getting excited about, oh my gosh, we can do all that. Maybe the assays in lab we're changing because we can paint something somewhere and the worms can crawl different ways. A lot of ideas are coming out of these collaborations. So there have been more collaborations in my lab. And one of them, I've had artists come to my lab for maybe 1 or 2 days, look at some images. This is a result of this. He's an artist, Jonas Angela. He took some of the images, some of the ones from Fleming, and put together a piece of work called Common Threads. And this past semester, I met a woman named Chanel Matsunami-Gavreau. She's a performance artist. She was in my lab all semester, and she had a project called Differentiate. So she looked at images of C. elegans cuticle of the worm. She found these green sort of designs, and she makes fabric and stuff for a lot of her performance art. So she made the fabric below in response to her experience of seeing the C. elegans. She also has sort of embryos in her hair that she takes out in some of her performances. So she actually was, my students had never like hung out with a performance artist before. I think they were just a little scared, but very interested, They were just, but I have that kind of, I like when these kind of people come to my lab. I learn a lot from them, and then Chanel's like, oh, I teach at Do It. She teaches how people use Photoshop. These kinds of, and my students are like, oh, I can ask you something, and we can talk about, my work, and they show each other work, and a lot of things come out of these collaborations. So, and Chanel's just been absolutely wonderful. I always think she's going to be the next Lady Gaga, I think, of performance art. She's amazing, actually. So very cool stuff. So some of the things I've been doing for many years in grad school is a lot of worm art. So when I was starting out in graduate school, I did this first sort of yin-yang of the C. elegans worm. It's quite abstract, but these are all logos for the International C. elegans meeting. Sort of what I became famous for initially is doing this artwork. I still do the logos. The one on the bottom with the 3 circles is the one for this year. And then I asked my advisor, John, if I can do an art show, because I did art shows with my parents all my life. So in 1997, he said, sure, don't do it, but I don't have time, you do it yourself. So I did. I set it all up, got everything working. So now if you go on the C. elegans page, it's the highlight. They want it every year. And they've been written up in a lot of science magazines, and people are always, and I'm doing one next week in LA, and people come from journals and interview me. So it's been very popular, and I think it's been great, at least for the scientific community, to tell the world of, here's all this taxpayer money, here's the beauty and the design for the public to see. I mean, this is what's coming out of this, and there's discoveries come from this kind of imagery. So I've been very fortunate to be a part of that, and I really enjoy it. So I was going to show you some images from this. So this is a gonad of the C. elegans. These are nuclei. And Abby is a professor. She's a Howard Hughes investigator at Berkeley. This is one of the first entries, the stained glass C. elegans. Again, all the people who have submitted for this, they all became faculty members. I say that there's a lot of creativity being a scientist. Here's a batik C. elegans on a scarf. That was one of the first years. Here's someone who got inspired by Andy Warhol. There's the actual images, the EM images, and made it up there. Some people find worms in nature. And my friend Kirsten said, I was walking around in the snow one day and I saw a worm. So they'll snap pictures. There's a lot of photography where they find worms and things. This is something that I did at Berkeley, is that sometimes in the plates that we grow, they get bubbles in them, and the bubbles actually look like little spaceships, and there's a little worm in the back there. So sometimes you see things throughout the day that are beautiful to you or interesting. Snap a picture anyway and save. It's not going to go in the journal, so I have huge collections of these things. So I always say that, all these scientists have done, the greatest scientists are always the greatest artists, and it's been the case for the art show, is seeing all these people who have entered, have all gone on to great things. There's that creativity is inherent. So then I was going to go talk about a little bit right at the end is some of my hobbies. Do things on my other sides beside my art. So I have a food blog called Food Scope, if you're interested in, and I make cakes. This was my wedding cake I made. So if I don't know how to do something, I learn how to do it. So I took 3 months in the summer before my wedding, learning how to make cakes and fondant and stuff like that. So I really like doing that, and I make wedding cakes for grad students. It's just a passion. So I sort of cooking is like home chemistry. So my undergrad professor said to me, you never trust a scientist who can't cook. So there's a lot of experimentation, and I agree with them. You got to know how to put things together and be creative, and miraculous things come out of these things. These are some of my I make scientific cakes. So these are zebrafish embryo cakes. So these got picked up by Nature last week on Twitter. Someone discovered that I had this on my website. So this is for my friend who got tenure. He works in zebrafish. So I do that. Those are the actual images down there. This one, 2 weeks ago, I had a paper came out. So these are mitotic spindle cupcakes from my student, Mary Kate. And I snapped a picture, and then I asked, I didn't tell my student, but I went and emailed the editor. I said, I attached this picture of these cupcakes I make. Would you be interested in posting it next to my scientific article? And then I ran in and told my student, she's like, you're crazy. They're never going to go for this. This is science. And I said, well, it doesn't hurt to ask. They're going to say no, fine. So they took it. They loved it. They emailed me back like 5 seconds later and said, I love this. So we're putting it on our blog. So it got, it's on the blog. It got out on Twitter, and then it got picked up. And lo and behold, Nature emails me and says, we'd like you to work for us as a guest blogger. So this is from Cupcakes. So, and my students were just like, you're crazy, but things, I said, that's what people want. People want, said, do what you feel passionate about, ask those kind of things. You'll never know what you're going to end up with. People think there's rules in science, but you can break them. And that's where discoveries are made, whether you're making cupcakes or not, or doing your science. And so, yeah, things, this has sort of been my life. I just do what I do. I don't try to be in that boundary or try to sit in that box of sciences to go out and explore different avenues, and it helps me. Making cakes helps me understand what I'm working on. You're constantly thinking about your work all the time in anything that you do. You don't have to be at work to come up with a discovery, those kind of things. My dad used to say Einstein used to sit out in the boat every day. That's where, bobbing around. Those are the kind of things where ideas come. So with that, I want to thank you and say I'm very happy to be here and give you this keynote presentation. Thanks.